Welcome to the first Lewis Wolpert Memorial Lecture. I'm Barbara Conrad, head of the Research Department, Cell and Developmental Biology in the Division of Biosciences here at University College London. CDB, as it is called, was Lewis Wolpert's home department until his retirement in 2004. As we will hear from Claudio Stern in a few moments, he remained a frequent visitor to CDB and the Stern Lab until just a few years ago. Lewis Wolpert sadly passed away January 28th. With today's event, we would like to remember and celebrate him and his work. Lewis Wolpert was a giant. With his groundbreaking ideas, he made conceptual advances that paved the way for modern developmental biology. And he inspired scores of young scientists and colleagues, many of them among us today. I never had a chance to meet Lewis Wolpert in person, but he certainly spoke to me through his textbook, Principles of Development. This textbook informed and inspired me as a postdoc more than 20 years ago. What I appreciate most about this work is its focus on principles, the important stuff, basically, rather than minute details. Lewis Wolpert was a giant. Maybe not surprising, today's memorial lecture has turned into a gigantic event. Not in our wildest dreams did we expect that we would get so many registrations, hundreds and hundreds of them. And right now, there are 492 people in the Zoom meeting and probably many more watching on CDB's YouTube channel. This is brilliant, and it reflects Lewis Wolpert's estimable standing within the scientific community. I also think that it reflects widespread interest in and excitement about developmental biology. These are great times for developmental biology. Through new technologies and techniques, we now can observe and interrogate individual cells in the developing animal. A dream come true. And this enables us to address stubborn fundamental questions that have remained unanswered to this very day. Lewis Wolpert's model for pattern formation proposes a two-step process. First, a cell determines its position within the embryo through a French flag-like mechanism. And second, that cell then responds to this positional information in a way that is dependent on its developmental history. Developmental history and what it entails is still not well understood. And this is just one of the many phenomena that remain to be dissected mechanistically and that can be traced back to an idea by Lewis Wolpert. A close colleague and friend of Lewis Wolpert for many years was Claudio Stern, Fellow of the Royal Society and the Academy of Medical Sciences, and JC Young Professor and head of CDB from 2001, when our department was still called Department of Anatomy and Developmental Biology, until 2014. A very distinguished developmental biologist himself, Claudio hosted Lewis Wolpert in his group for many years after his retirement. And this is how Lewis Wolpert continued to contribute to research activities until very recently. Indeed, Claudio just told me that Lewis Wolpert is co-author on a manuscript that he's currently working on. Now, before I hand over to Claudio Stern, who will present a tribute to Lewis Wolpert, some information about how the Zoom meeting will be organized. There will be no question and answer session after Claudio's talk, but there will be one after Cliff Tabin's talk. Until this question and answer session at the end, the audience will be muted and the chat disabled. Once the question and answer session starts, the chat will be enabled. And if you want to ask a question, please indicate this in the chat and I will invite you to unmute and you can ask your question directly. So with this, I hand over to my colleague, Claudio Stern, for his tribute to Lewis Wolpert, to which I look forward to very much. Claudio?
Let me unmute. Okay, so hopefully this will work. Let's just try that. I hope you can all see that. Okay, well, it's a very special occasion and a sad one and a happy one at the same time. We're very happy to celebrate Lewis, in fact, all together, and it's wonderful to see. I just had a quick look at the names and the profiles of the people that are logged on already or were a few minutes ago, and it's just wonderful to see so many friends of Lewis's and of mine and colleagues and others logged on all to share this, as well as some of Lewis's family. It's great to see you all here. So I'm going to give it a very quick introduction to Lewis for those of you who, particularly for those of you who've never met him, just to try and give a little bit of a flavor of him as a person and as a scientist and as a colleague and as a friend. And what better way than to use some of his own words in as much as I can. So I'm mixing up some anecdotal things, a little bit on science, a little bit on other things, and some of his own words as we go. Of course, that quote up there, it is not birth or marriage or death, but gastrulation, which is truly the most important time in your life, is probably known by most of our colleagues. Not everybody knows exactly why he said that, because of his work on gastrulation was really a very long time ago. He did it very early on in his career, but we'll touch on that in a minute. Let's start with Lewis himself.
Well, I was born in Johannesburg a long time ago in South Africa in 1929. Okay. And I was born into a Jewish family. And I was born into a family who wanted a nice Jewish boy. And I'm afraid they were a bit disappointed because I didn't turn out to be a nice Jewish boy.
So Lewis first studied engineering in South Africa, and he specialized in soil mechanics. But then he was obviously a rebel. He didn't want to be a nice Jewish boy, so his sense of adventure started to kick in. And he got a little involved in politics, partly by befriending friends of his own family. And he also met briefly Nelson Mandela. And in 1952, he set off with a friend, Norman Morrison, to travel through Africa, hitchhiking through Africa. And in their trip, they carried a letter from Nelson Mandela introducing them to various revolutionary or political groups as they traveled through Africa. And in that time, they spent 3 months hitchhiking. Here's a picture of Lewis here in a checkered shirt with a dead hippopotamus. And then they took an Arab dhow, which is a ship very crowded with people. These are actually real pictures. Here's Lewis wearing some interesting attire on the ship. And again, here you can see they were eating and sleeping rough on the ship. Eventually, they made it across the Arabian Sea to the Mediterranean, from where they went to Italy and France, to, I guess, a little bit cleaner than the time that they must have spent on that boat, but it must have been really, really exciting. So Italy and France, and from there he proceeded to Israel, where he had an offer to work on soil mechanics in Israel for a while. But he quickly got bored with that too. And so at that point, he got the opportunity to go to London. And this is Lewis again.
And then, because the man that I'd left behind, Jerry Jennings, who was so keen on me in South Africa, he had arranged that I should get a scholarship at Imperial College in London to study more soil mechanics. And I arrived in London in 1954 and went and studied soil mechanics at Imperial College. And it, of course, was bliss coming to London because nobody knew me. In South Africa, I had to behave in a reasonable way because so many people knew my family. In London, nobody knew me, and I could behave as I wanted to. And I had quite a wild time in Soho in the 50s. I've even got friends still from those days. It was very exciting.
Lewis made friends wherever he went. Whoever he touched, I think, always befriended him. The work on soil mechanics at Imperial didn't excite him either, really. So following advice from a friend from South Africa, Wolpert Stern, he moved across London to King's College. And there he joined James Danieli and started to do a PhD with Danieli. in working on the mechanics of cell division, where he could apply Lewis's engineering knowledge to biology. And so he tried to describe really the mechanisms of cell division and so on, and using a lot of microscopy. Immediately after the PhD, Lewis was offered a lectureship at King's. Those were the days you didn't have to do a postdoc always. You go straight from the PhD to a position, and then eventually, in a few years, promoted to a readership, like an associate professorship. During that time, he started to work on more developmental problems, like regeneration of hydra. This is one of the pictures from the time, from 1966, with Jerry Webster, who also unfortunately died very recently, about a couple of weeks after Lewis. And Lewis also initiated a very fruitful collaboration in Sweden with Tove Gustafsson, with whom he did a lot of work on gastrulation in the sea urchin. In fact, it's from this period that his interest in gastrulation stems. So again, working on the mechanics of cell mechanics of gastrulation, and really how cells are finding their way through the embryo to generate complex form and structure. This is what started to reveal to him really the complexity of cell behaviors in the embryo. This in particular that I'm illustrating here is that it's a wonderful review. It's 75 pages long in the International Review of Cytology from 1963, Gustafsson and Wolpert. It's a classic and it's still worth reading today. So those of you who are interested in this and haven't read this paper, it's definitely worth reading. It has a lot of very important principles. And it talks quite a bit about cell migration and cell interactions in very similar ways that we're now still asking. So a series of papers from 1960 to about 1967, 68 on sea urchins came out of that. Many, many very interesting observations there. In 1968, Lewis's work and engineering background together got him invited to a very exclusive meeting that had been organized by C.H. Waddington, the famous Waddington, in Italy, in Villa Sebaloni in the north of Italy, on Lake Como in 1968, where Lewis presented a paper that would soon become one of the most important and influential contributions of his entire career. And some of the people in this picture are really quite legendary in the field of theoretical and evolutionary and developmental biology. For example, René Thom, here, a French mathematician, and Christopher Zeeman, together were responsible for catastrophe theory and really interesting models of how you could explain the arising complexity in biological systems. Here's Christopher Longett Higgins, who's a psychologist doing theory, Waddington himself, John Maynard Smith, the famous evolutionary biologist. Here's Lewis. This is Donald Mitchie, who was Anne McLaren's husband. Brian Goodwin was also working on patterning in the embryo, and Lewis and Brian had a close connection. Brian Goodwin was actually my own PhD supervisor, so this is a very interesting collection of people from all walks of life looking at complexity and to try and arrive at what Waddington himself was trying to do, a theory of everything. So in that context, Lewis presents his work, his ideas at that time, on a principle that might explain how big embryos and small embryos can scale so that different numbers of cells, but appropriate proportions of the territory, are apportioned to different patterns, different fates in the scale of the embryo, whether it's large or small. So he used the analogy of the French flag. So Lewis's main idea here was not so much what most people know about, which is that the gradient and thresholds sets up the territory or scales the territory, but actually the concept that cells have position information. It's as if they know their GPS coordinates in space within the embryo, so they know exactly where they lie, and they interpret the local information to behave that way. And then the analogy to the French flag.
In fact, there's one particular concept that goes back to Alan Turing, who spoke about morphogens. and that is once again diffusible molecules that may be involved in pattern formation. And the real problem was, how could this gradient be set up, and what would it be a gradient in, and how would the cells really interpret it? But the key point with the French flag model was the concept of positional information. That is, that cells would have a positional identity, and that would determine how they behaved.
The same year, 1968, just 7 or so years after being appointed to his own position at King's, he got offered the position of full professor and head of the department that was called Biology as Applied to Medicine at the Middlesex Hospital in London, so across the other end of London. But he felt that hydra regeneration and sea urchin gastrulation were a little too far from medicine, so he decided to look for another system that he felt would be more suitable on which to work, and in particular, to try and test his ideas about the French flag and position information and patterning. And he chose the chick embryo initially, and in particular to look at what patterns the limb. So here's a chick embryo with the limb bud starting to grow out. I'm sure we're going to hear a lot more from Cliff about this. And in a normal limb, you have a pattern like this with the digits and its skeletal elements appropriately. And there have been some experiments done by John Saunders in America showing that if you do certain transplants here, you can get duplication of the digits and of the elements along one axis of the limb. And Lewis felt that this might be a good system in which to test ideas of morphogens. These are really quite difficult experiments to do at the time, and he was setting up and tooling up to do that. He took advantage of many very skilled experimentalists who were also actually had quite a lot of fun doing the experiments in the lab. And here's a wonderful picture. This is Cheryl Tickle, who's one of Lewis's long-term collaborators in the lab and now co-author of his Principles of Development as well, together with Dennis Somerville, who sadly also died a number of years ago, joining forces, as you can see, to do a four-handed experiment in one lab coat, to obviously having fun in the lab, but almost replicating the experimental result, as you can see. So they obviously had a lot of fun in the lab. Over the next few years, Lewis recruited a team of wonderful people who joined his group, many of whom went on to become very famous themselves. So here are Gerald Tickle, Amata Hornbrook, Dennis Somerville, Julian Lewis, Jim Smith, and his very long-term secretary, Maureen Maloney, who also died, unfortunately, a couple of weeks ago, and Nigel Holder, who was actually my direct predecessor as head of CDB, and he also passed through his lab. In fact, this is Maureen, Julian Lewis, and Nigel Holder in this picture. So he died prematurely in 1999, just 2 years after becoming head of department. So Lewis's great strength was, sorry.
I did. By the way, I never do an experiment. I am a theoretician. I hate doing experiments. My skill is to get other people to do the experiments.
So Lewis's great strength was the ability to ask questions, actually. Some could be quite naive. Some could be almost childish. Often they made you wonder why you hadn't asked it yourself. And most of them are still good questions today. But the value of his contribution was mainly in the asking itself, pointing exactly to what we don't know. So here's one example.
Could you all do me a favor, please? Stretch out your arms. How similar are they? There may be one or two of you that it's not the same. But just think that those limbs have grown for 14, 15 years since they were in the embryo, and they have almost identical length. And no one has a clue as to why that should be so. I published a paper on it. Nobody, not a single person, has come up with an explanation. Not one single person has come up with.
So he taught us that it is more important, as well as more fun, to ask good questions rather than to fill the literature with lots of data.
I'm afraid that in my own subject, developmental biology, there isn't a lot of work on general principles going on. And it's more about detail. It's just the gene does that, this protein does that. And I find that very boring. But God is in the detail, as they say. And I suppose in the long run it'll be okay. But for the short run, I find it a little tedious.
So the Middlesex Hospital and UCL merged in 1987. And in 1999, Lewis moved to the Department of Anatomy and Developmental Biology, which is now CDB, as Barbara introduced. And he often enjoyed, over those years, a game of tennis with members of the department, including Tim Arnett and former head of department Jeff Bernstock. And I was very fortunate when I came in as head of department in 2001 that Lewis became associated with my group. And for the following more than 15 years, we really greatly enjoyed his regular participating in our weekly lab meetings, where he always asked interesting and unusual questions, inspiring students and postdocs all the time. And again, just always probing. He was always interested in something. He was always finding a different angle and always doing it in a very, very fresh way. At the same time, in addition to his science, Lewis wrote many books, some to promote popular understanding of science, others about very personal issues that touched him very deeply, like depression. And he wrote these things both to try and understand it himself, as well as to help other people that were suffering with the condition. And he, in a very touching way. Here is a photograph of a particularly nice occasion, relatively recently, just over a year ago, when he celebrated his 90th birthday in 2019 at the Royal Society. And this is a cross-section of many developmental, British developmental biologists from the 70s and 80s that were connected to Lewis in some way. And I'm glad to see that many of those people are actually in the audience right now. And 10 years before, in his 80th birthday celebration, we hosted a little mini symposium in the department, in the anatomy building. And here, in fact, you can see Lewis looking at a book that we put together with Jim Smith to celebrate his 80th birthday. And right behind him is Cliff Tabin, who's our speaker today. And John Gurdon next to him, and Cheryl Tickle here in front. This is Cheryl, John Gurdon, and Cliff Tabin. And so we're all greatly saddened seeing these pictures and listening to him about Lewis's passing, and already miss him very much. So who more appropriate to give the first of what will be in future an annual Lewis Wolpert Lecture than Cliff Tabin? Like Lewis, Cliff started outside biology and studying physics in Chicago. He then moved to biology for his PhD, which he did with Bob Weinberg, where he worked on oncogenes and viruses. And then he went off to do a postdoc with Doug Melton in Boston as a postdoc to study embryos using molecular biology techniques. And then thereafter, he came to London for a short time. He wanted to move to more other systems where he could study limb development and patterning. And he went to visit Jeremy Brox, who many years also in our division in biosciences at UCL. At that time, he was in another building, but in Jeremy Brox's lab, in a few weeks in London, Cliff learned some techniques about how to get limbs to regenerate in newts. And then that got him started in the area of limb development and regeneration. Like Lewis, Cliff has a huge range of interests and a similar curiosity, and I think the same ability to ask fundamental questions that inspire others. I think Cliff is just extraordinarily broad. One of his many contributions is the discovery of and understanding of the function of the vertebrate hedgehog genes. And this wonderful picture of him with Lewis, I believe that what he has on his back is a hedgehog outfit. But maybe Cliff will correct me. If not, it maybe should be. It seems prickly. But they had some disagreements in terms of fundamental mechanisms of some aspects of limb development, but always friendly and constructive. And Lewis liked to remind people that that was the case, but that both of them were very close friends and at the same time debated in a constructive way about biology. So without further ado, I will stop sharing my screen and welcome Cliff Tabin to give the first Lewis Wolpert Memorial Lecture. You're muted, Cliff.
Sorry. Can you hear me now?
Yes, we can hear you now, Cliff.
Yeah, so somehow when you put up my screen, I lost the unmute button. I'm back now. So thank you, Claudio. That was just a marvelous introduction to Lewis. Yes, that was supposed to be a hedgehog outfit that I was wearing. There was this wonderful celebration in honor of Lewis, and Cheryl Tickle had gotten in touch with me and asked me if at all possible if I could arrive, because she was going to plan some skits, if I could arrive wearing a hedgehog outfit. And unfortunately, maybe in the UK you can purchase a hedgehog outfit. In the United States, hedgehog's not a native animal, and there were no hedgehog outfits to be found in any costume shops. So I asked my mother to make me something that would be a hedgehog outfit, and she took a rug and spiked the rug and sewed it to the back of a shirt. So that was my mother's version of a hedgehog outfit. Anyway, I want to start by just giving a few very short thoughts about Lewis from my own perspective. Lewis was obviously an enormous giant in the field of developmental biology, but he did impact my career at multiple stages in very important ways. And the first actually was, he was really my introduction to developmental biology. When I was a graduate student at MIT in the 1970s, they didn't even have a course in developmental biology. And I'd viewed developmental biology or embryology as a very descriptive sort of thing that embryos were obviously a very cool, very beautiful process, but not something that I really thought of as being something that there were principles that we could start exploring. And Lewis published a paper in the popular science journal, Scientific American, on pattern formation and developmental biology in 1978. And in it, he laid out his ideas of positional information and so on. And it really started for me to change my thinking. So I started to view developmental biology as a field of conceptual ideas, and then one that would be right for exploration at the level of genes. Now, I might have gone in that direction anyway, ultimately, because it is such an amazing process. But at the time, that article was really quite pivotal in starting my thinking about development in general and pattern formation in particular. Now, if you go forward a few years, I had a small lab and I was starting to do work with viral vectors, trying to understand something about limb regeneration and limb development. And the first time I had a chance to have an in-depth conversation with Lewis was at a limb meeting in Spain, in Santander in 1990. And meeting Lewis for the first time really was obviously struck by his enormous intellect and by his great enthusiasm for ideas. But what I actually remember most from that meeting was how warm he was. And that was really something that was very significant for me, because shortly before I'd attended a small American limb meeting in Ohio. And my first interactions with Americans were not so warm in the field of limb development. As an outsider who really had no developmental biology background, they gave me a rather cool reception as I tried to talk about the things I thought that one might be able to do with gene transfer into embryos and so on. At least initially. Ultimately, they became great colleagues and great friends, but initially there was a bit of a standoffishness. But with Lewis, he was exceedingly supportive, interested in what I was doing. He was just welcoming, which I very much appreciated. And as I got to know Lewis, it was really very much the way Lewis is with all colleagues, young and old. Subsequently, whenever we would cross paths over the years, I'd show him our latest data. And as Claudio indicated, we didn't always agree on some of the things that were going on in limb development. And his typical response to seeing my new data would be to reinterpret it for me. He'd ask me to sit down and say, oh, well, my boy, let me explain what your data is really telling you. He would try and tell me what my data meant. But part of it was because we really did disagree a little bit on some perspectives of limb development. But a lot of it was simply because he was a provocateur. He loved to stimulate an argument in order to get at the heart of a matter. And he would sort of push one, and we would have really marvelous conversations. And our interactions certainly made the science much better, gave us new ideas, gave my lab new ideas to think about and new directions to go. They were always stimulating and always fun. He was a spectacular colleague as well as just a great scientist. So in giving a talk in honor of Lewis, who always enjoyed seeing new ideas, I decided I wanted to present new ideas. That is not necessarily new big ideas. Lewis had big ideas, but at least new data that you wouldn't have seen before. So to do that, I'm actually sacrificing telling you a complete story. And instead, I'm going to tell you sort of two half-baked stories, two vignettes, one from the developmental side of my lab on limb development, which is of course the focus of so much of Lewis's work, and one from the evodevo side of my lab that includes a new context where we think a gradient of sonic hedgehog is very important, but not a gradient that provides positional information, but a gradient that's important in other ways. But although both of these are quite preliminary, it's all unpublished. I think it's the type of thing that Lewis would have been more interested in hearing than having someone rehash old data for them. So I'm going to tell you two stories today. One I'm entitling What Makes a Limb Bud Special? And the second, How Do Birds Evolve the Capacity to Vocalize? Now I want to set up the first one, that is to say the limb development story, in the context, in sort of in a Wolpert framework. So here you have, of course, the French flag model. There have been many drawings of the French flag. This particular French flag model pictures come from that 1978 Scientific American article that I mentioned before. It was striking to me when I was a grad student. Here on the left side is the idea of using the French flag to explain a gradient of a morphogen, something that Claudio already went over for you.
Yeah.
You need to share your screen.
What?
You need to share your screen.
Kidding. Okay, after Claudio's talk, it went up on my screen as if it was being shared. I'm sorry about that. We'll go back. I do apologize.
That works.
Hang on, almost with you. Okay, so you missed the earlier pictures, that's fine. So two short vignettes. What makes a limb bud cell special and how did birds evolve the capacity to vocalize? We'll start with what makes limb buds special. Here on the left is the French flag picture. Both of these come from that 1978 Scientific American article I mentioned before that I showed in earlier slides that you missed. It doesn't matter. This is, of course, the image that uses the French flag to describe a morphogen gradient. But what I want to focus on is the other panel that was side by side with this in that particle, which is the one over here, grafting between two different flags. Here you have a French flag and American flag. And what Lewis posited was that both flags were set up with the same type of positional information. But, hang on. There we go. So if you take the French flag before positional information is laid down, so this is a French flag primordium, and an American flag primordium before the information is laid down, and do a graph where you take this portion of the American flag that if left to its own device would eventually form stripes, but graphed it up to this corner of the French flag primordium. When positional information is laid down, it will know that it's in this corner. It won't think that it's down here. It'll get the positional information that says you're in the upper left-hand quadrant. But then because it's an American primordium, it'll make American stars rather than this solid blue field of the French flag. So this idea that there's a genetic context that positional information is sort of interpreted in is something that Julian Lewis and Lewis Wolpert published a paper in 1976 where they called this principle of non-equivalence. The limb primordium and the French primordium are not equivalent, but they both have positional information given to them the same way. Now, this was, of course, a theoretical French flag, but it was based on or congruent with embryological data. So, for example, in this classic experiment from John Saunders, Mary Gasling, and John Cairns from 1959, if you take tissue from a leg bud that left alone would form a thigh, this is tissue fated to become upper limb, graft it to the tip of a wing bud, presumably at a stage before positional information is laid down. Then positional information gets laid down. This tissue knows it's no longer in a thigh region, but it's in a digit region, but it doesn't make wing digits, it makes leg digits because, to say toes, because it has that underlying information. So again, this is this idea of non-equivalence. Now we have a sense now of where that non-equivalence comes from in terms of hind limb and forelimb. This is a picture taken by Malcolm Logan, and when he was a postdoc in my lab. Lots of labs have contributed to our understanding that at least among the genes that make a wing different from a leg, you have TBX5 in the forelimb, TBX4 and PITX1 in the hind limb. And if you're a limb that has TBX5 and not TBX4, then you know that you're going to make a wing type structure on the basis of your positional information. So there's a non-equivalence between the two limbs, but I'd like to suggest that there's actually three non-equivalences here, not just the two limbs, but there's a third one as well. And to understand that, let's take a step back and think about where limbs come from. So the limb bud, and this happens to be a mouse embryo picture, but the same would be true of the chick, of course. In cross section, a limb bud is basically a bag of mesenchyme. But you have to think about where that mesenchyme comes from, because if you go back a few stages from this sort of limb bud stage to a neurulus stage, there is no mesenchyme. The entire embryo is in fact epithelial. You've got an epithelial ectoderm, epithelial endoderm, epithelial neural tube, epithelial notochord, epithelial somite, epithelial intermediate mesoderm, epithelial lateral plate mesoderm, including both the somatopleural and splenopleural layers. No mesenchyme anywhere. The mesenchyme of the limb, which will be generated here, of course, arises by the process of epithelial mesenchymal transition, or EMT, where these cells will delaminate. And you can see that happening in both the forelimb and hind limb levels, where you have an epithelium that does not have any mesenchyme, generating mesenchyme in both the forelimb level and the hind limb level. The trick is, though, this isn't just happening at the limb levels. It's also happening in between. So you see here the generation of mesenchyme in the flank that ultimately will give rise to the dermis of the flank. And these three different mesenchymes, they're all made by EMT, all starting from lateral plate mesoderm, but they make different things. This limb bud, the wing bud, or the leg bud can make cartilage, tendon, bone, joint, dermis, whereas the flank will only make dermis. Now you could say that this is the reason for this is simply because the dermis, the flank mesenchyme is in the wrong environment. That is, the difference between this and this is not that it's not capable of making these different tissues, it's just not getting the signals telling it to do so. And indeed, initially, if you at first glance, there seems to be data supporting that concept. Classic experiments done by Marty Cohn and Cheryl Tickle's lab, and also by Sumihara Noji's lab, showed that if you put a bead soaked in FGF into the flank of an embryo, it would generate changes in developmental processes such that the flank, rather than just making dermis, now generates an entire wing. The trick is, both Sumihara Noji and Juan Carlos Espesa-Bamonte's labs took a look at when you can do this. And it turns out that this trick of putting a limb bud in only works until stage 16. After stage 16, this doesn't work. And that's significant because at the flank level, at stage 16, there's no mesenchyme. At the flank level, the process of EMT doesn't start till 17, 18. At the time the bead works, which is 14, 15, 16, you don't have mesenchyme, you only have the epithelialized lateral plate. And in fact, once the mesenchyme comes into, so while a number of FGFs can do this, Sumihara Noji's lab first showed that the proximal FGF that really does the trick is FGF10. If you put in a bead soaked in FGF8, it first induces FGF10, and then FGF10 induces the limb Well, once the mesenchyme forms, the FGF10 receptor isn't even expressed in these cells. The FGF10 receptor is expressed in the overlying surface ectoderm, as it is in the lateral plate epithelium before the generation of EMT. So it's not that FGF's not capable of converting this mesenchyme into a limb-like mesenchyme. And in fact, there's reason to think that this mesenchyme is fundamentally different. One way of showing that is by a different classic experiment called a recombinant limb. First technique I think was first developed by Ed Zwilling and really exploited to great use in a number of important papers from Marion Ross's lab. What you do in a recombinant limb is you take limb mesenchyme from the anterior of the limb bud at an early stage, unpatterned early naive mesenchyme. You dissociate it and repellet it. Then you take an ectoderm from another limb bud, could be a hind limb bud or a forelimb bud, and you combine the two, and then you graft it onto a host embryo. And if you do that, you actually get a reasonably good limb-like structure out of this recombinant limb. It's got a stylopod, a zeugopod, an autopod. It's not that well patterned, but if you give it just a source of anterior-posterior patterning, such as ZPA cells or a source of sonic hedgehog, and again, this is Marianne Ross's work here, you get a very, very acceptable wing-like structure. Well, you can do those sorts of experiments with mesenchyme from a wing or from a leg. And these are examples from my own lab. And I should say the wing recombinant and the leg recombinant look different, not because there's some fundamental difference between the wing and the leg, because this one was done by Jimmy Hugh and Kim Cooper when they were in my laboratory. And they were quite good at this technique. This was done, the leg example was done a little bit more recently by other people in my lab. But either the forelimb or the hind limb would work quite well in the recombinant limbs. But if you do that with the flank, you get nothing. You absolutely do not get, you don't get pattern, you don't get differentiation, you don't get tissue, you don't get anything. Now, one way of thinking, of explaining this could be with the same genes we talked about before. That is, one could imagine you need a TBX gene to make a limb. And we know that if you knock out TBX4, you don't get a hind limb, for instance. So it could be that TBX5 makes forelimb, TBX4 makes hind limb, and no TBX makes no limb. That would be one way of understanding the three non-equivalences. But alternatively, something else could be telling the cells you're a limb, you have the competence to make a limb, to be patterned, to differentiate in different tissues, or versus not. And that the TBX genes are layered on top of that to give these limb-specified tissues forelimb or hind limb character. So we decided to try and look and see if we could figure out the difference between limb and non-limb in the flank. And the approach we decided to take was essentially a reprogramming approach, sort of a Shinya Yamanaka approach, where we're going to take non-limb mouse fibroblasts, that is to say flank cells, and add a variety of transcription factors and see if we could turn them into limb progenitors that would be capable of making a limb. And because we're doing this in the mouse, we're not going to be able to do one of those recombinant limbs, but at least we can inject them back into a limb bud and say, can the cells that we transform contribute to limb development? And the reason we're doing it in the mouse is because in the mouse, we have very good markers for limb cells as opposed to flank cells. In particular, there's this transgene PRX-GFP that Malcolm Logan made that allows you to mark the limb bud cells as separately from the flank cells. And that's important in two ways. Number one, it gives you a marker to assay, are you successfully converting these cells into these cells? But more importantly, probably, in doing the experiment, we can use a cell sorter to make sure we have a pure population of flank cells or a pure population of limb cells for our comparisons. So the first question we had to ask was, can limb mesenchyme be maintained in culture? Because we're going to be doing the reprogramming in culture, and if you can't maintain legitimate limb bud cells in culture for an extended period of time, then you're going to be in trouble. And to assay whether we're maintaining them in culture, we'd use the GFP marker that's driven from the limb promoter PRX1, but also two other antibodies against two other limb markers, ubiquitous early limb markers, including SELF like 4 and LHX2. So if you use these three markers and you put limb cells in a dish, they'll proliferate for a while, but very rapidly after a couple days, all three of these markers, PRX1, LHX2, and SAL4, are very rapidly shut off. So we had to find conditions where we could maintain legitimate limb cells in culture before we could start thinking about reprogramming other cells into limb cells. And so we were fortunate at this stage that a master's student from France named Charlotte Cole came to the lab, and she took up the challenge of trying to get these cells to work in vitro. And she made enough progress that the initially leery people in my lab bought into the idea, so that when she returned to France after a year, a postdoc, Yuji Yatsuda, who did most of the work I'll tell you about, picked up the project. And the two of them together were actually able to find conditions that worked. I'm not going to go through all the permutations of how they got there, because that's not really the point. But just to summarize quickly what they had to do, there are two aspects to it. Number one, getting the limb bud cells to remain limb bud cells required moving them into 3D culture. For most of the experiments, we used a hyaluronan-based hydrogel because the limb bud itself is largely hyaluronic acid. But Matrigel, with commercially available Matrigel, will also work. So number one, you have to grow them in 3D. And number two, you have to add factors. And those include factors that we know are important, that are present in the early limb, that are anti-differentiation factors, as Roel Nusa and Jill Helms first defined, including Wnt, FGF8, and retinoic acid. And also required putting in antagonists of pathways activated during differentiation in the later limb bud, such as anti-BMP and TGF-beta and anti-Rho kinase to prevent differentiation from occurring. So if you have this combined set of conditions, now you can have, as opposed to serum where all of these markers are lost, here shown after 8 days in culture, all three of these markers, GFP, LHX2, and SEL4, are all expressed at a high level. You can see here in the quantification, they grow a lot better in this culture too. But besides growing better, you now maintain for an extended period of time all of these markers. So now we have an ability to grow the cells. Now we want to do a reprogramming experiment. We need transcription factors to put in. What transcription factors do you use? So graduate student Alan Rodriguez took up that task. And the first thing he did was he prepared RNA-seq libraries from forelimbs and hind limbs. But then he compared early forelimb and early hind limb stages. And then he compared those to both flank and neck. And if you do that, he got 1,654 forelimb and hind limb genes, of which 225 were overexpressed at least twofold in the neck or flank, of which 60 were arguably involved in transcriptional regulation in one way or another. Of those, some of them were expressed in differentiating tissues, like skeleton, for instance, but 35 had widespread limb expression. And then of those 35, we pared it down further by getting rid of redundancy. So for example, SAL1 and SAL4 both came in out of this screen. We just used one of those two. Or if HOX D13 and A13 were both in the screen, we just used one of those two. So we could get a more workable number. And we ended up finally with 18 non-redundant genes. So now here's the experiment. You start by doing a cell sorting to get GFP negative fibroblasts from the flank. So these won't have any limb contaminant. You put it into 2D normal tissue culture and infect it with the 18 viruses transducing the 18 candidate factors. After a couple of days, you add these limb-specific growth factors I mentioned before. And a couple of days later, you put it into the hydrogel with the limb factors and grow them for a couple of weeks and say, do you get putative limb-like cells? And the legitimate limb bud cells in culture form these very nice, large, round colonies. And you do not get those in the control experiment where you don't have the factors. But if you infect with these factors, indeed, you can get GFP positive colonies. They're not just GFP positive, but they also express these other markers like SAL4. Indeed, by RT-PCR, they express virtually every limb gene that you can think of. That you test, do they express MSX2? Yes, they express MSX2. They express MEIS2 and MEIS1? Yes, they do. So then we wanted to whittle down the list of 18 to a smaller list. And again, I'm not going to go through all the steps that we went through, but initially we did a dropout experiment where we said, if you remove any one of these factors, of these 18 factors, which ones make it less efficient? And then that gave us 7 factors. And then we took those 7 factors and gradually, by pairwise comparison of 2 and 3 factors together, again, without going through the steps that we took to get there, we ended up with a set of 3 factors: PRDM16, ZBTB16, and Lin28A. And these 3 factors together will maintain in flank mesenchyme the expression of all three of these limb-specific markers, GFP from the PRX promoter, LHX2 and CEL4, as well as by PCR, expression of any other gene that you want to look at from the limb. So it really seems like these three factors alone are capable of transforming a flank cell into something that starts to resemble a limb cell. Now you can ask, is it resembling a forelimb cell or a hind limb cell? So we've of course looked at those markers, and they seem to be forelimb in character. Without the virus, you don't have any TBX4 or 5 expression. With these three factors, PRDM16, ZBT16, and Lin28A, you can turn on TBX5 as you would in the forelimb, but you do not turn on TBX4 that you do in the hind limb. Now, the most likely reason is not because these three factors are driving forelimbness, but we suspect it's because we have retinoic acid present in the culture as one of the vital factors to maintain the cells from a very early stage. And by having retinoic acid around, retinoic acid acts on Hox genes and anteriorizes the early embryo. So I think it's probably, although we haven't done any experiments to show it, it's probably the role of retinoic acid to drive things towards TBX5 in a forelimb character. Anyway, besides looking at specific candidate genes, we also want to look at a transcriptome level. So another postdoc, Chang-Hee Lee, decided to do that. So in this slide, what I'm showing you here is there's no reprogrammed cells at all here. This is what you're just looking at the input cells. Here are the non-limb flank fibroblasts in 2D culture. Of course, if you put them in a different environment, they change their transcriptome somewhat. So here are the same flank cells in 3D culture. If you put legitimate limb progenitors from an E9 mouse embryo into culture, you get a different cluster. These are the mouse limb bud cells, distinct from the mouse flank cells. And then these are mouse limb bud cells that are put directly into the RNA analysis without ever being cultured. So putting them in culture does shift them somewhat. And this is E9, E10, and E11 limb progenitors directly out of the embryo. But these are limb progenitors that are treated the same way as the cultured cells in vitro. If you now overlay this with the expression analysis of the reprograms, oh, sorry, this is just verifying that these different clusters are what I said they are. So genes that are expressed in the flank but not the limb, like ACTA1, are expressed here. Limb bud cells that are not expressed in the flank, like LHX2 or NMIC or MSX1, are expressed in the limb clusters. Genes that come on with differentiation, like SOX9, are activated not in the early cultures, but only when you look at later stage limb bud cells. So that's the verification. But now when you overlay reprogrammed cells on top of this, they fall directly on top of the legitimate limb bud cells. There's really complete congruence of cultured limb progenitors and fibroblasts reprogrammed with PRDM16, ZBT16, ZBTB16, and Lin28A. So at least at an RNA level, they look like, the expression level, they look like limb bud cells. Do they actually behave like limb bud cells? So to look at that, we want to know, can they actually make cartilage, tendon, bone, et cetera, instead of just dermis? So first we tried to differentiate them in vitro. And indeed, if you put cells in addition, the absence of the factors that keep them undifferentiated, they start differentiating in all kinds of directions. Whether you're talking about limb cells or the reprogrammed cells, you get markers for chondrocytes like SOX9 or mature cartilage like Angrican. You get markers for tendon like scleraxis, markers for joint like OSR2, but none of these factors are upregulated in the unreprogrammed flank cells. So it seems like they're able to differentiate. Now, of course, a limb getting back to Lewis's ideas of positional information, you don't just need to be able to have cells that can make different tissues, but they have to make tissues in the appropriate places. Different segments of the limb have all the same cell types in the zygopod and stylopod, but the organization is different, the patterning is different. So to ask whether they can receive patterning signals, we want to look at some patterning genes, things that come out in response to positional information, like, for example, the Hox genes that are expressed in different regions that we believe are transmitting positional information. One in particular that we looked at is HOXD13. We know that HOXD13 is activated in response to both FGF8, which is distal, and sonic Hedgehog, which is posterior, giving you this distal posterior bias in HOXD13 expression. And if you take legitimate limb bud cells, early limb bud cells in culture, and add sonic hedgehog and FGF. So in this graph, all of the cells are seeing 100 nanograms per microliter FGF8, and you're adding either 0, 1, or 2 nanograms per microliter of sonic hedgehog. If you give them both factors, these legitimate limb progenitors will turn on HOXD13. Flank cells will not. Flank cells, not only don't they differentiate into limb cell types, they also don't respond to limb signals by turning on HOX genes. However, the reprogrammed cells do. So that's all in vitro. You can also look in vivo. In vivo, we want to mark the cells so that they stay marked when injected into a chick limb bud. So we mark different cells in slightly different ways. For the legitimate limb progenitors, we just took limb cells directly out of a mouse that expresses GFP everywhere. So every cell is expressing GFP. This is not a limb-specific promoter, and they'll continue to express GFP. So that marks those cells. You couldn't use GFP for the non-limb fibroblasts because first we had to select against GFP to make sure we didn't have any limb bud cells. So we had the PRX GFP to select against limb to get flank cells. So instead we transfected in M-cherry to mark those cells. And finally, we didn't want to just use the GFP from the PRX promoter for the reprogrammed cells because after they differentiate, the PRX promoter would go off. So we did a stop flock stop strategy where a PRX CRE would cause recombination of the flock stop allele, the stop flock allele of this otherwise ubiquitously expressed TD tomato. So that would give us a permanent marking. In any case, if you do that and inject them into limb buds, both limb progenitors and reprogrammed cells will remain in the chick limb bud. Flank cells from the mouse actually don't even stick around. Not only don't they differentiate anything, but they're completely lost. And you can say, what do they become? And really quite equivalently, limb bud cells or reprogrammed cells can become, for example, cartilage, here shown with collagen 2, both in the reprogrammed cells and in the legitimate limb progenitors, or tendon marked by collagen 1 in both the legitimate limb bud cells and the reprogrammed cells. So we really think that we're able to create limb bud cells out of non-limb bud cells. Now, one reason that you might think that that would be interesting, although it was certainly not our motivation, would be in the context of regenerative medicine. Other people have shown, this is from Gufo Lin's work, that if you take limb progenitors, they're able, and put them into a regeneration context, they're able to enhance regeneration. If you had any hope of doing that in terms of human regenerative biology, you'd have to have everything I showed you work with human cells. And it's not as easy an experiment because you don't have legitimate human limb bud cells to compare them to. But it looks like the same factors will reprogram human dermal fibroblasts into something resembling mouse limb bud cells. And when you inject them back into a limb, you can get some differentiation into cartilage. This is very preliminary, I don't want to make too much of it. But the preliminary indication is the same three factors do seem to work in the context of human cells as well. What I'm more interested in telling you about, though, is what these factors might be doing in legitimate limb. And as I said, this is a preliminary story. I don't have a lot of information on it. But interestingly, the factors that we've identified that could reprogram flank into limb, PRDM16, ZBTB16, and Lin28A, are not the factors that are found that we've already been defined as being involved in specifying limb fields. They're not, for example, TBX5 and TBX4 or HOX genes or any of the other genes that I, in fact, might have predicted would be the things that would emerge from the screen. So what are the genes doing? Oh, before I go there, I just wanted to say, of course, these genes do come on. I showed you before that if you reprogram the cells, TBX5 is activated. So either these three genes that we're talking about are upstream of HOX and TBX genes in endogenous limbs. That's one possibility. Or they're actually not normally upstream of them, but there's a positive feedback loop where these genes that we've identified are able to feed back and turn on the TBX genes in the experimental condition, even if that's not what they control endogenously, it's their primary function. So what are these genes? Well, Lin28A is not actually a transcription factor. It's an RNA binding protein. It has a very highly conserved role in regulating the LET7 class of microRNAs. And that's consistent with John Fallon's finding that LET7 microRNAs are present at a very low level in the early limb bud when Lin28 is expressed, but LET7 becomes expressed at a higher level at the same time as Lin28 disappears. So why would LET7A be important? Well, LET7, not in the limb bud, but in other contexts, has been shown to regulate a number to target, as in the degradation pathway. LET7 targets a number of genes that are important in limb development in other contexts, including HAND1, NMYC, CEL4, TBX5, ZBTB16. It also suppresses stemness factors, including OCT4 and SOX2 during IPS reprogramming. So the idea then would be that if Lin28 is there, LET7 would disappear, and this reprogramming factor that we found, Lin28A, would be acting by getting rid of LET7 to activate the expression of these limb-specific genes. So we don't know that that's the case yet, but at least that makes logical sense. A little bit more confusing are these other two genes. These are transcription factors, they're both zinc finger transcription factors. Neither ZBTB16 nor PRDM16 seems to be required to make a limb bud, in spite of my trying to argue that they're important for reprogramming cells into being limb character. ZBTB16 or PLZF has been knocked out. Maria Barna and Lee Neiswander analyzed it in some depth. They do have skeletal defects, but they certainly make perfectly good limbs. PRDM16 doesn't survive long enough to look at the skeleton, but they do survive long enough to know that they make perfectly good limb buds. The limb buds look fine. What's been described are craniofacial defects. So how would you explain that? Well, one hand-waving thing you can do is argue for functional redundancy. And we think that functional redundancy might actually be something to consider here, especially with PRDM16. There's a very close homologue called EVI1. They're both multipath zinc finger proteins. They both have the same N-terminal PR domain. They're very close paralogs. And they both affect gene expression through chromatin modification. And they have very similar functions in both normal and malignant hematopoiesis. But early during development, while PRDM16 is expressed both in the limbs and the craniofacial region, EVI1 is only expressed in the limb buds and not the craniofacial region. So it's perfectly consistent with it complementing or replacing the function of PRDM16 in the limb, so you don't see a limb phenotype, but its absence in the craniofacial region allowing craniofacial defects to occur. So we're going to go ahead and make the double conditional knockout and try and explore that question. For now, we won't really know for sure what these are doing in normal development, but we can say the PRDM16, ZBT16, and Lin28 are present in normal limb buds, but not trunk mesenchyme, not flank mesenchyme. And they are capable of imparting limb progenitor properties to non-limb fibroblasts. And at least we believe this provides an explanation, conceptually at least, an explanation for the non-equivalence in Lewis Wolpert's terms of the limb and the flank cells. So I don't have more data to present on this, but instead I'm going to shift gears and spend the next little part of my talk talking about a completely different question. And that's, how did birds evolve the capacity to vocalize? So shifting gears, I'm talking to you with my larynx. Most tetrapods, including amphibians, reptiles, and mammals, all vocalize with their larynx. Of course, the larynx, or your voice box, is the structure in your throat here, which has within it the vocal cords, these vibratory tissues, as well as a cartilage box that allows the voice box that protects the airway. Now, birds also have a larynx. It protects the inflow through the airway. They've got a perfectly good larynx, but they have no vibratory tissue. There's no vocal cords in the bird larynx. It may seem surprising, but birds have, for some reason, evolved an entirely different vocal organ called the syrinx. The syrinx is down here in the chest of the bird. When a bird tweets or quacks or squawks, its voice is coming from here, not from its mouth. And this also consists of a modified cartilage box and also has vibratory tissue in it. The name the syrinx, just as an aside, was coined by Thomas Huxley in 1872. Syrinx was a river nymph that was amorously pursued by Pan, and she asked her sister nymphs to protect her, so they turned her into river reeds. But she was still making these hauntingly beautiful sounds through the reeds instead of through her mouth, the sounds that Pan had fallen in love with. So he plucked the reeds and made the first pan pipes, or the first syrinx, which looks similar to the bird syrinx, and also make sounds like the bird syrinx. So the same hauntingly beautiful sound. And they do indeed make beautiful sounds. Here's the wood pigeon. And in contrast to it, here's the white-throated sparrow. And I'm playing these different songs to you because to highlight the fact that birds that make different songs actually have quite different syrinxes. The syringes of different birds are extremely diverse. The voice boxes of different mammals and reptiles are all pretty similar. The birds have an enormous variety of different arrangements of their cartilage and their vocal tissue and of the muscles that interact them. And the most interesting of all are the songbirds like the wood thrush. Well, that's a very complex song. It's a very complex song, and that song is made to complex because it actually uses vocal cords on the two different sides of the syrinx independently. That is, it can control separately the vibratory tissue here and here, so it can actually harmonize with itself. So it's simultaneously making sounds of different pitches, not just overtones, but completely different pitches on the two sides. So when and why and how did this thing evolve? Well, it's present in crown birds. All crown birds have a larynx and a syrinx, but their sound comes exclusively from the syrinx. Reptiles, as well as mammals and amphibians, have a larynx. They don't even have a syrinx. So the syrinx evolved somewhere in here. Now, whether it evolved in the context of two sound sources, did an intermediate have both a larynx and a syrinx? Or was there a silent animal that didn't have a larynx and didn't have a syrinx yet either? We don't know yet. And similarly, why did the syrinx evolve? If it evolved just in the crown birds, it could be related to flying, in which case it may have to do with the changes in the structure of the hyoid bone that the vocal tissue of the larynx attaches. It may have forced a new sound source. Or it could have occurred back here in a non-avian dinosaur, in which case maybe the lengthening of the neck had to do with some of the acoustic properties. And we're working both with a physiologist, Franz Goller, to try and explore some of those questions, and with a paleontologist, we're collaborating with a paleontologist, named Julia Clark, who's looking for fossil syrinxes in some of the tissue that we have of some of the transitional forms. She actually has, and she's published already in Nature, a syrinx from the Cretaceous, but that syrinx present in the Cretaceous was actually already in not just a crown bird, but definitively in a waterfowl. So we haven't yet found a syrinx from any of these intermediates. So we don't really know the question of why and how it evolved from the paleontological standpoint. But we can look at it from an evodevo standpoint. This is the evolution of novelty. You've got a bronchial tracheal airway that has a series of continuous rings of cartilage and no vibratory tissue. And you change them to make unique cartilage structures and insert vibratory tissue. So how did that occur? Well, we'll start with the cartilage and then I'll come to the vibratory tissue. One way to think about the cartilage arising could be that you'd make the same continuously patterned airway through a single mechanism, such as a Turing mechanism. And then after the fact, you somehow specify these as being different, and you enlarge them to form the specialized cartilage of the syrinx. Now, while that may make sense conceptually, there's a problem with it. The problem is as follows. While repeated patterns of cartilage rings could be established through a Turing mechanism, and I'll come back and explore that a bit more in a second, you have a problem at the junction, because you have Turing-derived, in principle, Turing-derived rings coming this way and this way and this way. What happens where they all meet? Well, what happens when they all meet, if you model this, is you get a mess. And in fact, that type of mess is actually what you see in animals that don't have a syrinx. So in a mouse, you have a very regular, not completely regular pattern of cartilages. You do have some branched structures, for example, here. The rings aren't perfect, although in birds they are. The rings aren't perfect, but they're pretty good until you get to this region here where all heck breaks loose. You can see it even more clearly in these. This is an interesting paper from 1891. It was an 1891 descriptive paper that consists entirely of the description of the cartilage at the tracheobronchial junction from 86 human cadavers. So these are not men wearing funky swim trunks. Up here would be the trachea, here are the two bronchi, and each one is different. The pattern, while they have standard series of rings above and below, where they come together, you get an erratic and non-reproducible pattern. So how do you get, and that's of course the opposite if you want. If you want to make a very specialized structure like the Searings, you want to do things at the junction that are exact and are controlled. So how's that done? So Evan Kingsley, a postdoc in my lab, decided to take this on. And the first thing he did was he looked at the timing of the formation of these rings. Now in the mouse or the alligator, which I'll show some of later, the rings all arise simultaneously. In the trachea, at the junction, and in the bronchi, you go from no rings to the primordium of rings to rings being present all at the same time. In contrast, in birds, they get around the problem of what happens in terms of interference simply by making the syrinx cartilages first. So here you have no rings, these large modified rings that'll form the syrinx form first, and then subsequently you get the cartilage rings above and below. So what's the difference? How do you get the timing here of these particular rings to come on first, which you don't do in other animals? So look at that. Evan did an RNA-seq experiment comparing the gene expression where the syrinx would be at the tracheobronchial junction in the alligator to the RNA expression in the rest of the airway. And then did the same in the chicken, the trachea versus the syrinx primordia, at a stage prior to the first stages of chondrogenesis. So he did RNA-seq and then said, what differences are there in the chicken between these two groups that are not present in the alligator? And what jumps out are, at the top of the list, hedgehog pathway genes, including sonic hedgehog in the mesenchyme and patched one in the endoderm. And they're expressed at a much higher level in the non-syrinx airway, in the trachea, than they are in the syrinx. And if you look at that, it's a temporal thing. So here, at an early stage, sonic is actually expressed throughout the airway. So this is a bit of a complex picture because you see the esophagus behind it. This midline expression is the esophagus that's behind the airway. But in the airway, you have continuous expression in the trachea, in the bronchi, down into the lungs. But then this expression shuts off where the syrinx is going to form, with continued expression just in the trachea and the bronchi. And then subsequently, it goes off in the trachea and bronchi as well. And the timing of that matches when these cartilages form. So after hedgehog goes away in the syrinx region, the syrinx cartilages form. After it goes away in the rest of the airway cartilages form. So is hedgehog really responsible for this? So to find out, we went to an in vitro culture system. Evan first showed that he could culture the airway of a chick embryo in culture for 5 days, and in vitro it would make the same cartilage rings and the same syrinx as it would in vivo. So that works. He then repeated that by putting hedgehog in, so at the time hedgehog would normally shut off. And if you put in a hedgehog control bead, the four cartilages of the syrinx form normally. If you put in a hedgehog bead on one side, the cartilage doesn't form on that side. Or if you treat the whole culture with a hedgehog agonist, SAG, you get no syrinx cartilages at all. And that's if you do it at an early stage. If you do the same experiment at a later stage, after the syrinx cartilages have already formed, if you then add SAG, having hedgehog everywhere, in that case, the syrinx forms, but the rest of the rings don't form. So hedgehog seems to be an antagonist. So to make a syrinx unencumbered by other patterning processes, what birds have done is they prematurely release this constraint of hedgehog. Hedgehog inhibits the formation of the cartilages. You release it first here, you form these rings first, and then you form the other rings. So how did birds come to this? So more accurately, why is sonic playing a role in the first place? What's sonic's role in the whole process? And a better idea of this came from looking at its expression not in whole mount, but in section. So in whole mount, I showed you that it's expressed throughout the trachea and throughout the bronchi, but it's not actually throughout the trachea. It's actually very dorsal. So here's hedgehog only in the dorsalmost aspect of the endoderm of the airway, both in the trachea and in the bronchi. And this is at a stage where it's no longer expressed in the syrinx, so it's already off in the syrinx. Patched 1, which is, in this case, it's the receptor, but it's also a marker for hedgehog signaling, is expressed throughout the airway, but in a gradient with, of course, the highest expression being near where sonic hedgehog is expressed, but enough hedgehog is made to diffuse to the bottom, and the entire thing is seeing hedgehog, so you're repressing cartilage all the way around. Now, how does this become important? Well, I told you before, Turing mechanisms can generate patterns, but they're actually, among the patterns they generate, it's very difficult for them to form parallel stripes. Tom Hiscock, working with Sean Magnuson, published in 2015 in the 2D simulations that one way to get a rather complex stripy thing into really parallel stripes is if you had a perimeter gradient of some sort. And you can, Tom modeled that for us. And there it goes. Okay. You can see that when you model that, it gives you a complex pattern, but it doesn't give you perfect horizontal stripes. To get the perfect horizontal stripes, he had to impose a dorsal ventral gradient of some sort. I'm sure we can close it. There we go. So here, if you have a relatively flat gradient, you get pretty parallel rings, although with some branching. Whereas if you have a very steep gradient, you get very well-aligned rings. So the prediction of that would be that there would be a gradient. This is just quantifying the gradient in the chick. So here's in the chick mesoderm, you can see that you can quantify this patch gradient. And interestingly, if you look at the mouse, you can also pick up a gradient, but it's pretty flat. And that correlates with the fact that in the chick, you have perfect gradients, and in the mouse, you have less perfect gradients, very similar to the predictions that Tom Hiscock had in the simulations. Well, if this is really what's going on, one can understand what this might mean by thinking about what would happen if this gradient starts to be shut off. The reason this works is initially you have a hedgehog gradient, you make a lot of hedgehog in the endoderm, so you get a patched gradient that covers the entire level of the airway, so no cartilage forms. As hedgehog starts to be downregulated in the endoderm, the gradient starts retracting. As it retracts, you have a region at the bottom of the airway, at the ventral surface, where there's no longer any hedgehog. At that place, you form the initiation of cartilage, and the reaction diffusion can very easily get you a series of dots, on, off, on, off, on, off. Then what happens is, over time, the gradient continues to retract. As the hedgehog is progressively shut off, you would have a larger and larger region where you don't have any hedgehog signaling. You'd have an extension of the cartilage until it goes all the way around. Now, if that's the way you sort of draw parallel rings from a initial starting point on the ventral surface, the prediction would be that if you looked at the development, you would see the start of the cartilages would be at the ventral side, the opposite side from hedgehog, and that at any stage of this, during this pathway, during this process, you should be able to see that the gradient is indeed retracting over time, and at any given time, the cells that are forming cartilage should be the reciprocal of those that are expressing patched. And all of those turn out to be the case. If you catch it just at the right stage, the start of the rings actually is on the ventral surface. You can see here that the rings are being initiated ventrally and not on the sides or dorsally. You can see that the patch gradient that initially comes almost all the way around here now comes less of the way around, less of the way around, less of the way around. Between hedgehog 33 and 38, the gradient is progressively retracting. And at any stage you look, with a hedgehog reporter here now showing you where hedgehog activity is, and now in this case, red is patched, in this case, red is SOX9, chondrogenesis is exactly the reciprocal of where there's hedgehog signaling. So the prediction of that would be that if you block hedgehog signaling uniformly at the time the gradient starts to retract, that should result in chondrogenesis starting in multiple places and irregular formation of rings. And that's exactly what you see. So if you have a control airway, you start to form these rings on the ventral surface. This is in wild type. If you add cyclopamine and block hedgehog signaling, in the absence of hedgehog signaling, now the rings, chondrogenesis starts not just on the dorsal surface, but also on the ventral surface. Indeed, on the side, this is sort of a 2D confocal slice through the image. If you had other slices, you'd see chondrogenesis is being initiated throughout the tube, and they no longer are making perfect stripes. The cartilage, the initiation points of cartilage no longer line up perfectly. And this process of having the rings retract and having the reciprocal of the places that are undergoing chondrogenesis versus the places that are actively expressing patched or seeing hedgehog signaling was present before the origin of birds. You see the same mechanism of patterning the airway in crocodilians as well. This is taken from alligators. So the argument here is that in the ancestor, in the alligator, as well as in birds, it's a gradient of hedgehog activity that initiates chondrogenesis in the ventral surface and allows perfectly parallel rings to form as the gradient retracts. And by starting the process of shutting off this gradient earlier at the level of the syrinx, you're able to make the syrinx cartilages without worrying about interference from the touring patterning of the rings throughout the rest of the airway. So the final thing I want to tell you briefly about is how similar are the syrinx and the larynx. They're both making sound. And indeed, Huxley, who gave the name syrinx, also called it the lower larynx. But is it just a larynx? So Charlie Longtine, a graduate student in my lab, took a look, and they have a lot in common. For example, if you look at what extracellular matrix proteins are expressed in the developing syrinx, and then now we're looking at the vibratory tissue, the soft tissue, the same sets of extracellular matrix proteins are expressed both in the syrinx, the vibratory tissue of a chick syrinx, and the vibratory tissue of a mouse larynx. But of course, that could be simply by convergence. You have the same mechanical properties. You need something to vibrate. You need to make a sound. You may have independently selected for the same gene expression patterns in the two cases. Or the thing could have evolved through process homology or deep homology. That is, there may be independently evolved structures, but you could have co-opted a whole network of gene regulation that makes soft tissue, vibratory tissue in the larynx, and redeployed it in a new location to make a syrinx. Or it could be true homology. It could be that the precursor cells that make the vibratory tissue of the larynx, instead of migrating to the neck, migrate into the chest. So we wanted to see which of these might be the case. We first looked at direct homology, and that was something that we were able to eliminate pretty quickly. John Wallingford's lab had done experiments where they fate mapped neural crest and showed that the vocal ligament or the vocal cords of the mouse are completely neural crest derived. So we can see whether the neural crest derived by a transplant in the chick, where you take a GFP labeled chick neural tube and graft it in before the neural crest is generated, and put it into in place of the tissue in a wild type chick. If you do that, you get a lot of neural crest labeled cells these are all neural networks. But the syrinx itself is completely free of neural crests. In fact, the syrinx itself is made, one can show, of lateral plate mesoderm. So it's not homology. It's totally different cell type embryologically that makes the vibratory tissue of the syrinx versus larynx. What about process homology or deep homology? Well, to look at that, you have to know something about how the expression of these different extracellular matrix proteins are regulated, which hadn't been looked at in either the syrinx or the larynx. To try and look at what the regulation might be, Charlie did an RNA-seq experiment, identified cells from the developing vocal cord. This is now not the cartilage or the airway, but this is the soft tissue of the airway. And you get a large number of different types of cells. You can identify some of them. Some of them are going to form cartilage, for example, here. But there's one group of cells that particularly stood out. And those are the ones that are marked here, RA. They express a large number of retinoic acid synthetic genes, shown here. They downregulate a number of different RNA antagonists, shown here in things that degrade RNA, not RNA, the retinoic acid synthetic enzymes and things that degrade retinoic acid. And they also express this full set of extracellular matrix proteins that characterize the vocal cords. And you can see in situ that indeed these are all correlating. Here you have any of a number of genes that are involved in synthesizing retinoic acid or represent retinoic acid activity, are expressed specifically at the level of the Searings. Genes involved in retinoic acid degradation, like CYP2826C1, are expressed through the airway, but not at the forming Searings. And the extracellular matrix is there at the Searings. This is true for multiple extracellular matrix proteins. Here, you're just looking at a graph comparing cartilage versus retinoic acid synthetic enzyme cells, and highlight to see the extent to which they express lumicin, fibronectin, or serpin-F1. These are all different extracellular matrix proteins that are specific to the Searings, and they all are specific to the cells that express retinoic acid. In addition to retinoic acid, another gene that came out of this was SOX9. SOX9 is, of course, expressed in the cartilage itself, in the cartilage rings. But in addition, outside of the cartilage rings, SOX9 is expressed very specifically in the forming Searings, right adjacent to the cells that are positive for hyaluronic acid. So we want to know whether these genes, retinoic acid synthetic genes and SOX9, are in fact important for the activation of these different genes. First, we can look at retinoic acid. Retinoic acid does seem to be upstream of both SOX9 and extracellular matrix, because if you add an retinoic acid inhibitor, AGN, you lose both SOX9 expression and the synthesis of hyaluronic acid. So both of those are diminished. However, if you add retinoic acid, it's not sufficient to turn these on. So retinoic acid, it does turn on the hyaluronic acid binding protein activity. That is, it makes extracellular matrix. But SOX9 is not activated by excess retinoic acid in the trachea, but in the Searings, you need retinoic acid in order to turn on SOX9. So we have retinoic acid is necessary but not sufficient for SOX9, and is necessary for extracellular matrix and sufficient for extracellular matrix. You can also ask whether SOX9 is playing a role. In this case, we use retroviral vectors carrying either going into the trachea, a retroviral vector that transduces SOX9. If you add SOX9 to the trachea, you activate hyaluronic acid binding protein activity. So your extracellular matrix genes come on when you add SOX9. And if you get rid of SOX9 with a dominant negative construct, if you lose SOX9, you lose the formation in the Searings of these extracellular matrix proteins. It's not just hyaluronic acid, but if you add SOX9, you also turn on Versacan, you also turn on elastin. So we have a pathway where retinoic acid turns on SOX9, and both of these act to turn on the extracellular matrix proteins. So now we have something of a pathway. We can go back and say, and this in principle gives a convenient way to, if you're going to want to make a whole set of different extracellular matrix proteins in evolution, you wouldn't have to independently select for each of the extracellular matrix proteins. If instead you could just co-opt the upstream regulation, you could turn on the whole set of extracellular matrix proteins at once. So the question is this process homology, or was there convergence? So if there's process homology, you would expect to have these same pathways active in the larynx. And we don't have any functional data in the larynx, but you can ask, for example, the crocodiles, are the same genes, retinoic acid and SOX9, activated specifically at the larynx as they are specifically at the Searings? And the answer is yes. So this is from an alligator, and both SOX9 and retinoic acid synthetic enzymes are turned on next to the forming vocal tissue of the larynx in the alligator. Similarly, in the mouse, you have here a comparison I showed you before in the chick Searings of hyaluronic acid, which is an extracellular matrix protein, and SOX9. You have exactly the same configuration in the larynx of the mouse. So although we don't have functional data, at least it's consistent with the idea that the pathway that makes the vocal tissue of the Searings was co-opted from the origin of the larynx. Now one can actually take that back a step further, at least in this sort of very hand-wavy just hypothetical sense. Where could the program that forms the larynx have come from? The larynx itself, this does not present in the fish, of course. The larynx evolved in early air-breathing tetrapods to protect the airway, and the laryngeal valves formed initially to help control that airway, and then were extirpated for vocalization, exapted for vocalization. So where could they have come from? One possibility is another set of floppy valves that you have in the body are the heart valves. And interestingly, if you look at the zebrafish heart valve, this is the chick heart valve, has hyaluronic acid binding protein and SOX9 in the exact same configuration. And that's also true in the zebrafish. In the zebrafish, experiments from different labs have shown that retinoic acid is required in heart valve precursors, and that SOX9 is actually upstream of hyaluronic acid and elastin, some of these same extracellular matrix proteins, in the formation of the heart valve, as we showed they are in the Searings. So although it's very hypothetical, it's possible that a program that first evolved to make the valves that control flow of fluids in the heart was co-opted to control the flow of air in the larynx, was then modified to be able to be used as a vibratory tissue in the larynx, and then co-opted to give you the vocal tissue of the Searings. So how did birds evolve the capacity to vocalize? Well, we're starting to get a handle on a couple aspects of it. Through altered timing of sonic downregulation, it allows them to form these very specific cartilages unencumbered by the patterning of the rest of the cartilage. And then the co-option of the vocal fold program from the larynx was used to be able to construct a syrinx that would perform the same function of allowing vocalization that other animals are able to do with the larynx. So finally, just to thank the people who did the work. The work on the reprogramming of the limb cells was largely the effort of Yuji Yatsuda. The RNA single cell analysis from Chang-Hee Lee. The identification of the transcription factors was Ellen Rodriguez. Getting the whole project up and running and doing the in vitro culturing was from Charlotte Cole. And I do want to acknowledge that a very similar project was started at exactly the same time independently by Ernesto Lujan in Olivier Perquet's lab, who's also in my own department at Harvard. And when we realized we were working on the same thing, we sort of collaborated and had a very consistent communication between the two groups. The work on the Searings was the work of Evan Kingsley and Charlie Longton. The modeling that I showed you is from Tom Hiscock. We've collaborated with Julia Clark and Franz Gohler. Julia's the paleontologist and Franz a physiologist to try and understand other aspects of the evolution of the Searings that we haven't gotten to the point where I have stuff to talk to you about today, but we're working on it. And finally, I want to give a big shout out to Mike Dyer at St. Jude Hospital. So Evan Kingsley's wife, about 2 years ago, got a job at the University of Mississippi as a historian. And they had a young baby. So he didn't want to leave his baby, but he didn't want to leave his postdoc. And what he did was he moved down to Mississippi, and very nearby in Tennessee is St. Jude Hospital. Mike Dyer was kind enough to give him a bench and to give him the materials he needed to work with. So Evan's actually doing his postdoc in Mike's lab. Mike's lab is a mouse lab that works on cancer. He doesn't work at all on chicks. He doesn't work at all on evodevo, but he was very generous in letting Evan work there. And without that, we wouldn't have had the work that I could show you today. So I'll stop there and I'd be happy to take questions. Thanks.
Thanks very much, Cliff. I've been asked by several people that we probably shouldn't have questions because it's running quite late and some people are leaving. So if you don't mind, I think maybe some people might want to do this by email or whatever if they're interested. So it's a shame, but people have other things set up. I just want to make a final thank you and an announcement. First of all, thank you to Barbara for hosting it in the department, and especially to Roberto Mayor as well, who had the idea of doing it very quickly and got this all sorted. And the idea is now that this will become an annual lecture that we will host, a Lewis Wolpert Memorial Lecture that we'll have every year thereafter, except next year it will be a bigger event. And I'm very happy to announce, here's a poster, very happy to announce that next year we're going to have a symposium that will be a 2-day symposium that is co-organized by Jonathan Slack, Jim Smith, Gerald Tickle, Neil Vargas, and myself. It will take place hopefully in person and hybrid at the same time, ideally on the 13th and 14th of September 2022. And so next year we will not have the lecture, we'll have the symposium instead, and thereafter it will restart with lectures annually in memory of Lewis. So I hope to see many or all of you there, virtually or ideally in person. There'll probably be space for about 100 to 120 people physically, and we hope that we are going to be able to do that then. So thank you all very much, and thank you, Cliff, for a wonderful talk. Always, as always, very inspiring. Thanks very much. Bye.
And I would also like to thank Claudio, actually, and the Division of Biosciences for making this possible, and also Michael Wright and Adam Shallot, who really helped make things happen today. I think it was wonderful. Both presentations, I'm inspired, I learned a lot. And as Claudio said, we hope to see many of you again next year and in 2 years from now. Thanks all for coming. And yes, it's been a great event. Thank you.